Every multicellular organism begins its life as a single cell. To see how that unit multiplies into trillions of others, we have to look down at the microscopic scale, inside a cell preparing to divide. This is mitosis, the specific process where a single parent cell divides to produce two daughter cells that are genetically identical to the original. During this equational division, the chromosomal number, represented as 2n for a diploid cell, remains constant, ensuring no genetic information is lost in the transition. Mitosis, or the M phase, is specifically the mechanical separation phase. It occurs only after the cell has already duplicated its DNA during the synthesis, or S phase, of interphase. The entire mechanism of mitosis is designed to solve a single logistical problem: how to physically split a cell while maintaining an exact copy of the genetic blueprint in both halves. Before division can begin, the cell must change the physical organization of its DNA to prevent it from tangling or breaking during transport. In a resting cell, DNA exists as chromatin, a loose, dispersed mass. To prepare for division, this material coils and condenses into the dense, structured units we recognize as chromosomes. Because the DNA was replicated earlier, each condensed chromosome is composed of two identical halves, known as sister chromatids. These sister chromatids are held together at a structural pinch point called the centromere. On the outer edges of the centromere, the cell assembles two protein discs called kinetochores. These serve as the attachment sites for the fibers that will eventually move the DNA. Understanding this chromosomal architecture is necessary, as these structures provide the physical framework for the mechanical sorting in the next phases. Cell division follows a two-part sequence. The cell first performs karyokinesis, the division of the nucleus, before it can execute cytokinesis, the division of the cytoplasm. Karyokinesis begins with prophase, the longest stage. The nuclear infrastructure dismantles, the nucleolus disappears, and the envelope breaks down, exposing the chromosomes. Meanwhile, duplicated centrosomes move to opposite poles, radiating microtubules to form asters and construct the mitotic spindle. By the end of prophase, the cell has transitioned into a polarized, structurally organized environment ready to manage its genetic payload. In metaphase, the mitotic spindle fibers attach directly to the kinetochores on each chromosome. Once the connection is established, these fibers exert equal and opposing tension from both sides of the cell. This tension pulls the chromosomes into a single line along the central axis of the cell, known as the equatorial plate or the metaphase plate. Because the chromosomes are at their most condensed and clearly aligned state, metaphase is the phase scientists use to study chromosome shape and number under a microscope. This central alignment serves as a final mechanical check, ensuring that every chromosome is properly positioned for an equal split. Anaphase is the shortest and most active phase of the cycle. At this stage, the previously aligned chromosomes pull apart and move toward opposite poles. This separation is triggered by the simultaneous splitting of the centromeres across every chromosome in the cell. Once separated, sister chromatids become independent daughter chromosomes. As kinetochores pull them toward the poles, the centromere leads, causing the chromatid arms to trail behind in distinct V, L, or J shapes. At this moment, the cell has successfully achieved its genetic goal. By separating the identical chromatids, the cell has doubled its functional chromosome count to provide a full set for each new nucleus. Telophase reverses prophase's structural changes. Arriving at the opposite poles, the compact chromosomes unwinding, returning to their stringy chromatin form. A new nuclear envelope reforms around each DNA mass, and the nucleolus reappears, establishing two distinct nuclei. While karyokinesis is finished, the cell is still a single unit with two nuclei. It must now perform cytokinesis to divide the rest of the cell. Animal cell membranes pinch inward, forming a cleavage furrow that deepens centripetally until the cell severs. Plant cells have rigid walls, so they divide centrifugally. Central vesicles aggregate into a cell plate, growing outward to create a new dividing wall. The completion of cytokinesis marks the end of the M phase. The result is two autonomous diploid cells, each carrying an identical set of genetic instructions. Looking past the individual cell, we can see how this process supports the survival of the entire organism. First, mitosis is the primary driver of multicellular growth and development. A human adult begins as a single-celled zygote. Building that body requires millions of mitotic divisions to produce the necessary cell population. Second, it allows for cell replacement and tissue repair. The body uses mitosis to replace damaged or dead cells. This includes the constant renewal of the skin, the gut lining, and blood cells, which have a naturally short lifespan. Third, mitosis maintains the nucleocytoplasmic ratio. As a cell grows, its volume increases much faster than the size of its nucleus. As the cell gets larger, the nucleus struggles to manage the expansive cytoplasm. Division restores this balance, allowing the cell to function efficiently. Finally, mitosis provides the genetic stability required for asexual reproduction in single-celled organisms and vegetative propagation in plants. Mitosis ensures that the same genetic instructions are conserved as a cell population expands. These equational mechanics provide the baseline for cellular life before the introduction of genetic variability in meiosis.